everybody. Today I am going to demonstrate the estimation of serum insulin using ELISA. So first of all, I will demonstrate the standardization of the ELISA kit. So here we are using the DRG Insulin ELISA Kit. As we know, insulin is the principal hormone responsible for the control of glucose metabolism. It is synthesized in the beta cells of the islets of Langerhans as the precursor proinsulin, which is processed to form C-peptide and insulin. Both are secreted in equimolar amounts into the portal system. Then the mature insulin molecule comprises of two polypeptide chains, chain A, which is of 21 amino acids, and chain B, which contains 30 amino acids. So the two chains are linked together via the disulfide bridges. So the secretion of insulin is mainly controlled by plasma glucose concentration, and the hormone has a number of important metabolic actions. So the principal function is to control the uptake and utilization of glucose in peripheral tissues via the glucose transporters. This and other hypoglycemic activities such as inhibition of hepatic gluconeogenesis, and the glycogenolysis is counteracted by the hyperglycemic hormones including the glucagon, epinephrine, also known as adrenaline, growth hormone, and cortisol. So insulin concentration is severely reduced in insulin dependent diabetes mellitus and some other conditions such as hypopituitarism. Insulin levels are also raised in non-insulin dependent diabetes mellitus, obesity, and some other endocrine dysfunctions such as Cushing syndrome and acromegaly. So the kit we are using here, this DRG ELISA kit, is based on the sandwich principle. So we have got this micro titer plate, which is coated with a monoclonal antibody directed towards the unique antigenic site on the insulin molecules. Then we have this enzyme conjugate, which is an anti-insulin antibody conjugated with the biotin. And then we have the enzyme complex, which includes secondary antibody, which contains streptavidin peroxidase enzyme complex, which binds with the biotin anti-insulin antibody. And we have this wash solution. It is of 40x concentration. So this is the stock solution. Whenever we perform the experiment, we have to make the working solution of 1x concentration. Then we have this substrate solution, which is the substrate for our enzyme. And then we have this stock solution. At last we have to add this stock solution to stop the reaction. Apart from these reagents, we have some standards like standard 0. So its concentration is 0 micro IU per ml. Then we have the standard 1, which is 6.5 micro IU per ml. Then we have the standard 2, 12.5 micro IU per ml. Standard 3, 25 micro IU per ml. Then standard 4, which is 50 micro IU per ml. And then we have lastly standard 5, which is 100 micro IU per ml. So before proceeding with the test, first we have to standardize our kit using these standards. Then we can proceed with the test procedure. So now we will start performing the standardization. So this is the guide which we have got along with the kit, and this is the protocol written here, the test procedure. So we will follow this procedure to perform the standardization. So the first step is to add the standards or the sample, 25 microliter in each well. So now we are taking the standard 0, 25 microliter, the standard 1, the standard 2, standard 3, standard 4, and the last standard 5. Now we will add 25 microliters of enzyme conjugate in each well. And we'll mix and incubate it for 30 minutes. Now we'll proceed to the next step, in which we will wash each well with the wash buffer. So for this we are taking 300 microliters of wash solution of 1x concentration in each well. So we have to do 3 times washing. This is the first time. Discard the wash buffer. And discard the wash buffer. And the final third wash. We'll tap the plate on absorbing paper or filter paper to remove any droplets. We'll make sure that there is no droplets left of wash solution into the microtiter well. Then we'll proceed with the next step. Now we'll add the enzyme complex 50 microliters in each well. And then we'll again incubate it for 30 minutes. Now we'll again do the washing. We'll discard all the contents and then we'll again wash with 300 microliters of the wash buffer. And this is the last washing. Again, we'll shake slowly and discard all the contents. and then we'll again tap the microtiter plate against the absorbing paper. Then we'll make sure there's no droplets left of wash buffer in the microtiter plate. So our plate is completely dried. Now we will add 50 microliters of substrate solution. Now we cover this plate and incubate it for 15 minutes for the color to generate. We have incubated it for 15 minutes and you can see the color has been developed. The color is directly proportional to the concentration of insulin. So we have taken 6 standard. From standard 1 to 6 the concentration is increasing. So we can see the color is also getting darker from top to bottom. So now we'll add the stop solution to stop the reaction and then we'll take the reading. So we'll take 50 microliters of the stop solution in each well. So as you can see, the intensity of the color developed is clearly visible. So we'll also take the OD in spectrophotometer and we'll make the standard graph according to the OD that we'll get.